a 300 to 600 pound fish goes for high quality sushi is $100 a pound. So that fish on the Japanese sushi market is worth $30,000 to $60,000 for one fish. So right now, tuna populations are just crashing as a consequence. And here you can see this is just one, haul on a Japanese market where people go to buy these tuna. And so they're just basically driving tuna toward extinction. So you guys should think about that when you eat tuna. Okay? Yeah, I'm not sure what's going on with albacore. But I like tuna, but, what are you going to do? Okay. So now here's one more fish, the opah, which was recently discovered in 2015. I'll send these various papers to you after today so you have them to study over the weekend because you're going to have a video on Thursday, so chill. And then your exam is next Tuesday, so you have a whole week to study for your exam. And I'll send these papers to you after today, okay? At any rate, so this is the opah, which is a pretty interesting fish. And again, here is the temperature graph in an opah, right? And you can see that there's really high temperatures right around its head. And what these guys did was they measured temperature profiles in an opah. So here's the fish is released. The black line represents the depth at which the fish was swimming. The blue line represents the temperature of the water at which the fish was at. And then the red line represents the pectoral muscle temperature of an opah swimming at depth. And you can see that that temperature is relatively constant across, okay, relative to the temperature that we see in the water itself. So this actually is one of the only known endothermic fishes, or even homeothermic fishes, a fish that can maintain its body temperature, not just regional heterothermy, but its entire body much warmer than the surrounding environment. And the way it does that is it has a rete mirabile system just outside the gills. So it runs warm blood next to cold blood as that blood, oxygenated blood, comes out of the gills itself. And so here's the rete mirabile with blue and red blood vessels, where the red, I believe, was cold and the blue is actually hot, so kind of vice versa. And the rete is located in the gill arch itself, within the gill, and it's also surrounded by fat tissue to act as an insulator. Here's the gill filaments, and here's the arch. The rete is actually surrounded by adipose tissue to enable it to be insulated, to keep that heat, right? And then here's a cross-section of how close these vessels are running to each other. So this is an efferent artery and an afferent vessel, right? Afferent means it's leaving. Afferent means it's coming in. Efferent means it's leaving. And these vessels are running right next to one another, so there can be heat transfer. And you can see just how densely packed those vessels are in that rete. And so that enables, right, the fish to basically warm the blood before it goes to its entire body, and therefore elevating its entire body temperature at a higher rate. Pretty cool, huh? So these retes occur in all these different locations, and it's all based on this concept of concentration gradients, whether it's oxygen or whether it's heat, right? And that concentration gradient flows in that countercurrent mechanism. And there's what a real opah looks like. It's a pretty ugly fish, isn't it? Pretty cool, though. These youngsters caught it. I bet you put up a pretty good fight. Right? It's a pretty cool fish, though. Right? All right. Okay. So now let's go to pressure. Right. So we talked about O2 concentration, and then we talked about temperature. So now let's go to pressure. So with regards to pressure, water is really dense, so you don't need any weight-bearing bones. Organisms could get bigger, and so consequently, because of the buoyancy of water, a 460 kilogram tiger shark really only weighs 3.5 kilograms in the water. It's pretty light, right? So organisms can get larger. And you remember, if you get larger, then you flick your tail and you can move a longer distance. It's an advantage to you if you're a predator, blah, 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 right? So there's some advantages to being larger. Oil also floats in water, and the way sharks handle this is they have oil in their livers, which makes them very buoyant, right? Because oil floats. So other fish don't do that, though. Other fish use, so this was, this is the OPA, right? And the OPA has endothermy. Right? So these are all the cases where the RETI, right, the counter-current exchange mechanism, is involved in all of these different mechanisms, okay, and also locations. So now let's talk about a swim bladder. So teleosts typically have a swim bladder, and they have a couple different types of swim bladders. The Physostomus is one where there's an air duct. That's what pneumatic means. An air duct between the gut and the bladder. They gulp air, and then that allows them to put that into their swim bladder and help control their buoyancy, because air is lighter than water, right? But others have this thing called a physoclistus swim bladder, and this is talked about on pages 75 to 77. And in this case, another rete mirabile is involved. Another counter-current exchange mechanism is involved in the swim bladder and in controlling buoyancy. Right? Oh, sorry. BU. Sorry. Made a mistake. BU. Buoy. Buoyancy, like a buoy. Okay? Okay, and then sharks have oil. We just mentioned that. All right, so how does this work? So this is kind of cool too. So that's a Physostomus one, where the guy basically gulps air, and then there's a duct that leads into the swim bladder, and then they can control loss of that. So this is depicting what kind of fish? What does it look like to you? Yes, carp. Okay, so carp, that's the way carp deal with it, right? So you think of an animal that's going to be living in shallower water, has access, lives along the surface. But in other fishes where they have this physoclistic type of swim bladder, they have a rete mirabile, or a gas gland, that occurs that allows them to magnify the concentration of oxygen in that gas gland, and therefore control the movement of oxygen into the bladder, even if the bladder already has a high concentration of oxygen in it. Right? So they can go against the oxygen concentration gradient by creating higher oxygen content within the gas gland, and then that gets through osmosis or diffusion, goes into the swim bladder itself. And then basically that oxygen gets absorbed into the vessels, and they use these muscles in this ovale region to control loss of oxygen, and that's how they can control the amount of oxygen that's actually in the swim bladder, so then they can control their place in the water column. Okay? And the way this happens is by two different factors, the Bohr effect and the Root effect, and I would encourage you to look these up both in your book and wherever else you might find some information. But what happens is we're looking at the level of hemoglobin saturation that occurs in the blood, and when we say hemoglobin saturation, that's the number of oxygen molecules that's bound to hemoglobin, right? The higher up, the more oxygen is bound. And the Bohr effect represents what happens when you get blood near tissue that is already metabolized and is giving off CO2. Okay? When you give off CO2, CO2 binds with water. Anyway, CO2 binds with water and forms carbonic acid. Actually, it's bicarbonate first. So it binds with water, and that forms H2CO3, which is bicarbonate, right? And then this dissociates into H plus and HCO3 minus, and that's basically carbonic acid. And in fishes, this is catalyzed by carbonic anhydrase, excuse me. That's a protein, basically, that converts bicarbonate into carbonic acid. And when you create carbonic acid, when it dissociates into this, what are these typically called? What's that? They're typically called protons, not protons like in the nucleus. But what does that do when you have free H plus ions floating around? What are you doing? You're creating an acid which does what? Lowers your pH. Right? When you lower the pH, what happens is you're causing hemoglobin. This is a shift called a Bohr effect, which you lower the pH, creating a more acidic blood. That causes oxygen to be released from the hemoglobin. And that oxygen then is what's going on here in the gas gland. You increasing the acid concentration lowers the pH. That lowers the affinity of hemoglobin for oxygen, and it releases oxygen into the gas gland. So that's one thing that happens. Another thing that happens is something called the root effect, which is related to this. And it's not only just a lowering of hemoglobin saturation, but it also influences what's called the stoichiometry of the hemoglobin molecule, and that prevents the hemoglobin molecule from having a high affinity for oxygen. And so once it loses oxygen, it doesn't want to pick it up again. And that also enables more oxygen to be released. And so you can create more oxygen going into the gas gland, even though there might be a high oxygen concentration in the gas gland already. And that's how you can, those processes allow the fish to control how much oxygen is going in there.
It's almost like cyanide poisoning.
Well, cyanide binds to hemoglobin, doesn't it?
Yeah, but it prevents it from taking up oxygen.
Yeah, well, probably because it binds to the sites, yeah. In this case, this is more what they term an allosteric interaction. And what goes on there is in the root effect, you're actually changing you change the structure of the hemoglobin when you bind to it, and that prevents the hemoglobin from binding to the oxygen by changing the three-dimensional structure of the molecule itself. Does that make sense? So normally, like our hemoglobin can hold 4 oxygen molecules. They bind to these heme groups, these iron groups, and it's because of the shape of the protein that allows that binding to occur. When you have a root and Bohr effect, what they end up doing is that changes the shape of the hemoglobin molecule, preventing them from having an affinity for oxygen and causing it to release oxygen. Right? So kind of cool, right? Isn't this stuff cool? Allosteric interactions and concentration gradients and physoclistus and physostomus, and doesn't that just like get your hair on the back of your neck standing up, you know, and you're thinking about, yeah, I'm going to go home and I'm going to talk to my wife about that, and she's going to tell me, yeah, whatever. And but when you talk to your significant other, they're going to be like, wow, you're smart. You were going to ask me a question.
So the Bohr and the root effects, so they only happen if it's physoclistus?
Well, no, that's what happens. I don't want you to get that impression. The Bohr and root effect happens in fishes, but the Bohr effect happens in you.
No, but I mean if it's physoclistus.
Yeah, but, well, instead of physostomus. Yeah. That's the mechanism behind it, right? But that mechanism still occurs in other places in your body. It just happens to be amplified in that gas gland because, again, we're having these vessels that run right next to each other that can establish these concentration gradients between them. It's almost like you can start down here and you have a concentration gradient of oxygen, and as you go across these vessels, that concentration gradient gets amplified. So that REAT gets amplified. Okay, at any rate, I think this is fun stuff, but, you know, that's maybe just me. I don't know. Okay, so it would behoove you guys. How many of you guys had a physiology class? Who'd you have it from? Oh, but you haven't had it in college yet? Yeah, do you have Dr. Lopez's class? He teaches cool class, doesn't he? You know, he's probably going to leave us. It's unfortunate. He's a great guy. I wish he'd stay. Okay, So now we talked a little bit about movement, right? We've talked about this before. So this is kind of a rehashing, or it's going over it again. But we talked about how they have a fusiform body shape. They have these epaxial and hypaxial muscles, placoid scales and elasmobranchs, and then you also have glenoid scales and tenoid scales and stuff in teleost fishes. They all do the same thing, that it's there to reduce drag, right? And so you can flow through the water better. Okay, so you guys should know that. Okay, so now let's talk about osmoregulation. You got one more? Oh, no, sorry. Okay. So now in osmoregulation, we've talked about now concentration gradients, but now what we're going to talk about is the diffusion of salts in water across semi-permeable membranes. And keep in mind that you want to keep your body solute concentrations at a relatively constant level for your body to function properly, right? And if your concentration gradients go too high or if they go too low, then your proteins and other processes in your body aren't going to work as well. Okay? So there is a kind of a nice area where it's kind of, you're maintaining homeostasis, if you will. So how does this happen? Well, for us, we've got this great thing called a kidney. And within that kidney, we've got something called a nephron. And in that nephron, we have things like Bowman's capsule, and the glomerulus, proximal distal tubules, collecting ducts, loop of Henle. These are all things that allow us to set up concentration gradients to absorb salts and or water in the kidney. And of course, this is where urine is created, right? Okay? So you guys all have these in your kidneys, and they're functioning for you right now. And one of the things that's happening is as your arteries enter, so this is your glomerulus, it's like this network of arteries and whatnot, and you have, actually, not arteries, I should say arterioles, so very, very small, like a capillary. At any rate, you have one coming in, that's your afferent, one coming out, that's your efferent, but in here it's like kind of a net, and because of the pressure, your blood pressure, it's pushing fluid out through the semi-permeable membrane of your arterioles into this Bowman's capsule, and then that fluid is going down into the rest of the structure of your kidney, and that's called a filtrate, and that fluid then is being either, you either put water into it, or you take water out of it, or you put salts into it, or you take salts out of it, and that's how you're maintaining this concentration of salts in the rest of your body, right? And certain things influence you, hormones influence that. So for example, vasopressin is one hormone that influences your water uptake. Alcohol is something that's called a diuretic. It causes you to lose water, right? So that's why you pee copious quantities of dilute water when you drink lots of alcohol, right? It's causing your body to get rid of it. Right? So, figure 410, see that? Talks about your glomerulus. Okay, so now let's look at what happens with different fishes with respect to concentrations of solutes within water that these fishes live in. Okay, so first, you have to think about that the body surface of a fish is impermeable to water. You should be writing these things down in your notes, for example, table 4.3, page 85 to 89. That's a note to you. That's where you can get more information, right? It's in your book. Okay, so their body surface is impermeable to water, but the gills have to be permeable because of gas exchange, right? That's where oxygen and CO2 exchange is occurring, right? So your gills are permeable, and so therefore the gills are subject to all of these physical and chemical processes of concentration gradients and loss of molecules across a semi-permeable membrane. And so what happens is that different fishes do different things to handle living in certain environments. So a hagfish basically has a solute concentration inside its body that is equal to the solute concentrations in its environment, so in the salt water. So there's no loss or transfer of solutes very much in a hagfish, right? Okay. However, in fishes that live in freshwater, they have higher internal solute concentrations. That means they have more salts in their body than are in the freshwater. So freshwater fishes have more salts in their body than outside, right? And marine fishes have less salts in their body than outside. And so that creates two different problems for these fishes, right? So how do they handle this? Sorry, you don't necessarily have to know this, but halene, where does halide come from? What does that mean? Chemistry? Salt. So steno means you have a very narrow range of salt concentrations that you can tolerate. Uri means broad or flat. You have a broad tolerance to salt concentrations. So that all depends on how your body deals with these different concentrations, okay? So now we're going to look at freshwater bony fishes versus marine bony fishes and how they deal with these different situations, okay? Freshwater bony fishes now live in an environment where they have more salts in their body than the surrounding environment. So what does their salt want to do? The salt wants to stay in the body? It wants to leave the body because of diffusion gradients, osmosis, right? It wants to leave. Water wants to come in. So they don't drink water. They create large volumes of dilute urine to get rid of the water that comes into their body, right? But in contrast, marine bony fishes are just the opposite. There's more salts on the outside of the environment than in their internal environment. So salts want to come in and water wants to go out. So they actually drink seawater to get water, but then they put salts into their urine to get rid of the salts that they drank. Right? And so let's take a look at that at another figure, which is here. And why does that occur? That's because, again, the gills are permeable to the movement of ions and water. So in a freshwater teleost, your body fluid is around 300 millimolar, right? So that's solute concentration. Remember moles? Moles is what? How do you calculate moles? What is a mole? A mole of anything. Avogadro's number, 6.02 times 10 to the 23rd molecules per mole, right? Okay, so this is 300 millimoles, right? And your body is impervious to water, but your gills aren't. So what happens is water wants to go into your body, right? Because you live in a freshwater environment. Okay? But then what happens is you want to keep salts. Salts want to leave your body. So what you do is you actively pump salts out of the water through your gills. So you have sodium chloride pumps in your gills that take salts out of the water, even though it's a little bit of salt. And then in your kidneys, you actively pump salts out, so you create lots of dilute urine to get rid of the water that you brought in, right? And keep the salts in your body, okay? So again, this is figure 412. When you get into the saltwater environment, it's just the opposite. You have less salts in your body than in the environment. So water, you lose water. Consequently, you want to get rid of salts as well, right? You're losing water, you're becoming too concentrated. You drink water to get the water back, and then you retain some salts and you get rid of other salts. So it's like this kind of balance. Your kidney is keeping some salts in your body while your gills are actively pumping extra salts out of your body in order to maintain that balance of being right around 350 millimolar concentrations in your body. Okay? So read that stuff. It's kind of interesting. You should be interested because you're a biologist. You're a wildlife biologist. And some of you are fish squeezers. How many here are fish squeezers? 1, 2. Oh, a minority. I love you guys anyway. No, I like fish a lot, actually. I just don't know them as well as I know birds and mammals. But fish are cool. And Dr. Colley's like one of my good buddies, so he's taught me a lot about fishes and stuff. Yeah, TW.
Why is it that bull sharks can swim in fresh water?
Good question. Chondrichthyans. What do they do? I gotta get rid of that noise. Remember the whole idea about urea and TMAO maybe having contributed to them secondarily losing bone as an endoskeleton because urea may break down bone and cartilage isn't as sensitive? Well, this is how they deal with increasing the concentration of solutes now. We're not talking salts in some respects. We're talking about molecules that are dissolved in water. That's what a solute is, right? They retain urea and trimethylamine oxide in their blood, and they don't drink water, but this raises the solute concentration of their blood so that they get around that concentration gradient, right? Because now they have dissolved solids in their blood, right? And so that's the way sharks deal with it. Now, some sharks can live in fresh water, and they still retain that urea, just not as much, right? So they control osmotic regulation by putting this protein back into their blood. So they have a different mechanism. Okay, so there we talked about osmoregulation. Where else are we? Let's go on. Doggone it, I got to get rid of that stuff. All right. I think I probably say this every year and I never do anything about it. Okay. So now we're going to talk about the final little bit about sensory systems. 11:45 to 1. We got to go, don't we? So don't worry so much about vision. Don't worry so much about taste. Let's talk about mechanical receptors and electroreception, these last two concepts, which are kind of cool. This is the lateral line system that runs along the side of fishes. It consists basically of this structure called a neuromast cell. It has a gelatinous cupula, which is like a top. And then these kinocilia are cells that are connected to nerves. And when they're pushed one way or another, that causes a nervous impulse. And so the fish basically can feel the pressure, right? And that moves these neuromast cells, exciting the nervous tissue, and that's how the fish can sense pressure along the side of its body. And obviously, it's an extremely efficient mechanism given how they can move through the water and whatnot. And then finally, we have these things called ampullae of Lorenzini. Now, if there is ever a term that is going to make your significant other think that you're smart, it is going to be the ampullae of Lorenzini, because that just has a nice ring to it, doesn't it? Ampullae, like ampule, right? Something that you hold that has liquid in it. Okay, and Lorenzini, he's the guy that discovered this. But it's a gel-filled canal, i.e., gel-filled ampule, right? And it conducts electricity. And so this can pick up the ionic imbalance that's created by ions flowing across your cells, which creates that little bit of an electrical field that all of us give off, but that we can't feel, but that these fishes can detect. And they can use the ampullae of Lorenzini to detect other organisms. And so, for example, in skates and rays, they can detect organisms that are buried in the sand by going over them, right? Sharks, when they go to bite you, at the last minute, they roll their eyes back so they can't see you, but the ampullae of Lorenzini tell them, yes, my prey item is right here. All I need to do is open up my mouth and chomp, etc., right? So really cool little structure that is found primarily in the chondrichthyans.